All right, the second part of Chapter 5, looking more at digestion of lipids and looking at LDL and HDL, some of the lipoproteins. So the first review here is what enzyme is responsible for digestion of triglycerides. I think you know that from the Chapter 3 slide, but here it talks again about lipases in your mouth, your stomach, and your small intestine. So there's a small amount of lipase, actually gastric lipase, made by the chief cells, which are cells lining the stomach. So the short chain fatty acids can be broken slightly in the stomach as well as in the mouth. And then the final breakdown from the small intestine is breaking down long chain fatty acids, but most digestion occurs in the small intestine. So it's all lipases, every enzyme. It's just a matter of identifying where the source of the enzyme is. So what are the end products of fat digestion? That we already talked about, how we can have a monoglyceride or a diglyceride or a triglyceride. So as fat is broken down by the enzymes, it becomes a monoglyceride and free fatty acids. And that's how it can be moved into the enterocytes, into the cell. So fat is broken down, and then as our body digests it, we have bile acids that help emulsify that fat so it can be absorbed. And there's also a hormone. This hormone, there's another one too, but cholecystokinin or CCK, that is stimulating. So there's things that trigger the release of these different lipases. So CCK, cholecystokinin, means the hormone is going to produce the pancreatic enzyme as needed. So then that's the byproduct. And then another hormone that might be essential. Well, here's more about cholecystokinin. So it's a gallbladder movement. So it's what the cholecysto is, gallbladder. So not only does it stimulate the enzyme secretion, but it also delays gastric emptying, so not too much food moves into the small intestine too quickly. And it also helps regulate our appetite, because as we fill up with food, it kind of triggers your brain to say, we've had enough. So there's quite a few actions for that hormone. And then here is the bile. So the bile is the other part that helps the digestion. So as we have these triglycerides, they can easily be surrounded by bile acids so that the lipases can interact with the different fats. So it breaks the large droplets into smaller droplets, as we saw earlier, and then these little lipases can interact and break down the fatty acids from the glycerol before it's absorbed. So that's another action of the enzymes. And then another question was, what are the differences between the absorption of long versus short chain fatty acids? So we haven't mentioned this too much, but we talked about how a fatty acid, it can be is a hydrocarbon, it's a long chain of carbons, but the chains vary. In the same way that you could have double or single bonds, you could have more than one double and more than one single bond. You can also have different lengths of amino acids. So the short chain fatty acids, long chain fatty acids. So absorption is different depending what kind of fatty acid. In general, the way that fat is carried, as we mentioned, is the phospholipids surrounding, making this micelle so that fat in the middle can easily be transported. So that's the transportation, and absorption is what's called a chylomicron. So it's a chylo meaning, I don't know why, fat, micron, a small portion of fat. So this moves into in our blood, from our liver and our lymph. You'll see these chylomicrons, which are a type of lipoprotein. There's some proteins in here as well. You can see here the protein molecule, the triglyceride inside, there's cholesterol inside, phospholipid along the outside, more cholesterol mixed in with a phospholipid. So it's just kind of a conglomerate, like a snowball. When you think about a snowball, all the things it can pick up if you roll it down a hill. So it's picking up proteins and fats. So that is the short and chain. But how do they digest if digestion change? So they're absorbed. It turns out that the short or medium chain fatty acids are absorbed more quickly because they are shorter, and they require a little different way of absorption. So here's different chains, like this just shows a shorter, a longer chain. So lauric acid, they each have a name, is a longer chain fatty acid than caprylic acid, for instance. So when it's absorbed, most of the fat is absorbed by just diffusing through the cells. But because short and medium chain fatty acids are more water soluble, when they have the phospholipid attached to them, they can go right into the portal blood system. They don't need to go into the lymphatic system. So the long-chain fatty acids, when they go into your cells, reform the triglycerides, and then they have to be transported by a chylomicron. So as you saw here, inside this droplet are triglycerides. Well, they break down, they absorb into the cells, and then they reform into triglycerides, and then they come together in these chylomicron combinations to move into the lymph system. So the chylomicrons do move through the lymph system, but the shorter chain fatty acids actually go directly into the blood. And so it provides very quick energy. So that's a little different in how those two absorptions occur. And here's another picture of how this is really helpful because it shows the large droplet, the breaking down of the micelles with the bile acids mixing in, allowing it to be hydrophilic. And then it can break down to all these little pieces which can easily absorb into the cell of your small intestine. So now you're in the lumen of the intestine, now you're inside the cell of the intestine, and you can see that these long-chain fatty acids reform. They mix with the glycerides to reform into triglycerides. So why are they doing that? Because that's really hard to transport. Well, it's because they don't transport directly as triglycerides, but they mix with the phospholipids, the proteins, cholesterol, and form this lipoprotein, and that can travel in the lymphatic system. Whereas the short-chain fatty acids have a much quicker system. They go right to the portal vein, to the liver, and then there they can mix with to form other types of lipoprotein or just to be used for energy. So that's the difference in how those are transported. Another section of this series is about the lipoproteins. And now we're talking what are the functions anyway of lipoproteins. You've probably heard of LDL, HDL, which are lipoproteins. So these are the fats packaged in the small intestine and transported, as we just mentioned. What is the difference between, what are the different types of lipoproteins? Well, here's the bigger picture of a lipoprotein, and then you can see that there's different types. So you might have a chylomicron, which is what we've just been discussing, but then depending what's inside this, they have categorized these. So if your snowball has picked up a lot of triglycerides and not very much protein, so just looking at the types of constituents of the lipoprotein will tell us what it is. So a VLDL, very low density. So I always think of density, low density. Fat is a low density because it always floats on water. So a very low density lipoprotein has higher fat content and lower protein. A very high density lipoprotein will sink because it has protein and less fat. Very low fat and very high protein. So why do you think it's more helpful to have high levels of HDL and low levels of LDL? Because this just tells you when you have a lot of HDL in your system, you have lower fat. If you have a lot of LDL, it's slightly lower in protein, has more cholesterol, a little higher triglyceride. So you can just see that the constituents change. So that is how we just differentiate, and then how they're packaged. So when you eat something with fat, say you had an avocado, so then the chylomicron can deliver, after it's packaged in the small intestine, to take the fatty acid to the cells as needed. But from the liver, where you've stored, remember we talked about how insulin will take extra glucose if we don't need it in our cells and store it in the liver. Well, if you have a mass of glycogen in the liver, it eventually turns into fat, adipose cell, and that can be then packaged as a very low density lipoprotein and delivered to the cell. So what are you going to do? It's not going to continue to sit in your liver forever. You have to move the fats that you've stored out of the liver as needed. So they're packaged as lipoproteins and delivered to the cells. On the other hand, when you have too much, what are you going to do when there's too much cholesterol, too much fatty acids maybe in a cell? So we have to have a way of clearing them out. So the HDL, the high density, the reason it's high density is it's picking up some extra proteins and cholesterol, taking it back, made by the liver and the intestine. So it is still made by the intestine, but it's much lower levels of cholesterol. And it's the scavenger cell, that will actually pick up the pieces of leftover cholesterol and to return it to the liver. So the scavenger cells remove LDL from circulation if there's too much. And these are some of the things that can cause the buildup of plaque, which we'll look at in a minute, and can be deleterious in causing congestive heart failure. So those are the way fat is transported in the system. And then the next section just asks us, like, what are the functions then of lipids? So what is the point of it? Well, we need triglycerides for energy. We also need it for the functioning and the cells of many different organs and tissues in our body. The phospholipids, those are mainly found in the cell membranes and transporting in the lipoproteins, transporting fats. And then some of the compounds that our cholesterol is used for, as we mentioned earlier, was bile acids, vitamin D, and also sex hormones. So estrogen, progesterone, testosterone are also very high in cholesterol content. So those are the source of the uses of fat. And here you can just see why we have fats for energy, as we mentioned. So it can carry vitamins. So if you have a low-fat diet, you could have a vitamin deficiency of these fat-soluble vitamins. We know that it's high calorie, 9 calories per gram. It provides good texture. So if you've ever had a muffin that's made with low fat, it's kind of chewy. It doesn't melt in your mouth. It doesn't have the same, what do you like about biscuits? What do you like about croissants? There's a lot of fat in between the layers of starch. It also helps with satiety, makes us fuller faster, obviously, because it has extra calories. And then it insulates and protects, so you have a layer of fat all throughout subcutaneous, right around underneath your skin. It insulates you, and it's good storage of energy. It's a very efficient storage for energy. So those are some of the reasons we have, the necessary reasons for fat. And you can see the phospholipid again in the cell membrane. It's structural. It has to be there. If this breaks down, your whole cell commits suicide. So we need extra fat for that purpose. I think the last series then is just this one here. We talk about how does percent of calorie in fat compare in America. So if you look at how much fat we get, the recommendation is to get 20 to 35% of our total calories, less than 10% from saturated fat, or even 7 to 10%. The average American intake is about 33%, so it's within that range, but at the high end, and 13% saturated, which is above the 10% recommended. So those are the things to cut back on. And you can see cholesterol as well is kind of within range. So the last section is the health, just the understanding that it's important for our health to try to maintain not too bad ratio of fats and not too much fats, but enough of the right kind of fats. So to prevent cardiovascular disease in the narrowing of the arteries, it's possibly from inflammation, from getting too much omega-3. It's possibly caused by extra LDL and VLDL. So what can occur is the normal artery with an injury can get this cholesterol plaque building up to a point where it blocks the blood vessel. So the main risk factors to try to eliminate would be keeping our cholesterol level, preventing smoking, keeping our blood pressure down, and then trying to prevent diabetes, because that does increase your risk for cardiovascular diseases. So the best way to lower your LDLs, reduce saturated fat and cholesterol, increase mono and polyunsaturated, and fiber. This fiber will help carry extra cholesterol out of your body. It actually attaches to the cholesterol and improves its clearance. And then the other thing is to try to raise your HDL by physical activity, at least 45 minutes a day, 4 times a week, eating regularly, and then less total fat. And then, of course, alcohol does increase moderate intake, they think will increase HDL. Other research lately has said that no alcohol is best, so it's still out what's the best thing for that. And then lowering triglycerides, just avoid overeating in general. Now we're talking about limiting alcohol, so there's some balance there, and simple sugars as well, including fish and small frequent meals. So those are the points for this chapter for review, and I think that our time is up. Thank you.